Namaskar दोस्तों, welcome to channel Toxicology on board. Today in this video we will discussing about a DABT 2022 list of questions topic based on memory. These questions I got it from LinkedIn. So let's see what are the tentative topics which the previously appeared DABT aspirants for 2022 they have got. So this is nothing but DABT 2022 list of questions topics based on memory. So first one was about kerosene burning in room in winter causes. We need to identify the toxicity gas name. Second is uh, greater than 10 micron particle size and deposit region. ADH inhibitor in ethylene glycol poisoning. So always important topic one important topic is about lung toxicity and the particles of that chemical uh, in which part of the lung they deposit based on the particle size, whether it is alveolar region or on the deeper parts of the lung. Next topic was ADH inhibitor in ethylene poisoning. So glycol poisoning is also one of the favorite topic for the DABT questionnaires. So ethylene poisoning, their symptoms we need to understand. We need to read in detail. Next, genotoxicity battery and significance. I have always discussed that why the importance, what is the significance of a battery of genotoxic tester. When it comes to systemic toxicity or carcinogenicity, there is no battery of test. But when it comes to genotoxicity, always we will be doing battery of genotoxic test. What is the reason? I have already discussed in one of my previous videos. Please kindly go through. In ISO, next topic is ISO 10993 categorization of endpoints testing based on. So as you can see, as per recent update in ICH Q3E guideline, there is the extractable leachables guideline which is under uh, like it is being circulated by the ICH committee for the comments from the industry. So in that also there will be discussion of extractable leachables. Similarly, ISO guidelines also there is a recent revision in the first ISO guideline in which I think that is 17 I guess, in which US has given no voter. So please such a recent trending topics also examiners will ask. Next topic is antidote for acetaminophen. So coming to paracetamol, that is acetaminophen toxicity, liver is the target organ. So what are the antidote? We need to understand the mechanism through which acetaminophen is causing liver toxicity and what is the best antidote for that. So one of the options you will have in that. Next topic is numbness, dizziness, case study because of what fish poisoning. So they will give you a case study like they will give you a question that one of the few two to like four members of family went to a restaurant and they were consuming a fish and suddenly one of the person has shown symptoms like numbness and dizziness. Then you have to identify what type of in that four options they will give you type of fish or some other seafood they might give. Then you have to identify which is the fish which is uh, causing this type of symptoms. Next question is about in silico toxicity, target prediction, both structure and ligand based. So one question you will definitely get on in silico technique because the recent industry is moving towards alternate methods. So in case of alternate testing, in silico methods like Derek, Lassa, as well as SARA, these are the softwares which FDA has also approved. So data generated from this software can be used for regulatory submissions. Particularly, this type of softwares are software prediction data is used for impurity qualification and also cosmetic industry as well as extractable leachables also. Next topic is methemoglobin. So in this, they I might have asked about iron state, the whether it was in Fe plus 2 or Fe plus 3 state. You need to remember hemoglobin structure, okay, how the light chains and heavy chains are binding, so how it is affecting the oxygen carrying capacity, deoxygenated oxygenated blood. So all these concepts related to hemoglobin, methemoglobin, you need to remember, and the symptoms also like blueness is there, the face like that you have to remember. The next topic is sigmoidal graph, potency and efficacy based. So from the Clayson second chapter, that is principles of toxicology, you'll get one question on dose response curve. So in that the different shapes will be there. like shapes like a S shape or J shape, U shape, inverted U shape. Okay, based on this, against what dose as well as drug response, efficacy or ear toxicity. So you need to remember in detail what is this um, type of gov. So one question in 2023 I appeared for the exam. So one question they will give you graphs and they will ask you to identify the type of graph. What that graph significant inference from that graph. Next is question number 11. One topic was on QA statement for GLP studies. So GLP, whether is the OECD GLP, important component in the case of preclinical studies, non-clinical animal studies. So GLP has various pillars are there in the test facility management, QA, study director, study personnel, TICO, archive. So with respect to their functions and responsibility, one question you might get. So one question in this year they got about QA statement. QA statement, which is a very important statement in all study protocols or study reports you will come across. So four options will be there. You need to identify what is that QA statement. Next topic was most effective route to avoid first pass effect. So one question on pharmacokinetics, AUC, Cmax, plasma. So they might ask, so they might give you some examples with respect to, uh, so you need to identify which of the following drugs will avoid first pass meta effector, or they will ask you about the route of administration through which you can avoid the first pass effector, like uh, parental route, like this one of the options might be there. Next topic was clinical route. 8 weeks correspond to same duration in non-clinical studies. So one question with respect to ISH M3R2 was asked. So you have to in one of my videos I have already discussed about ISH M3R2. So in M3R2 guideline you will have some tables. So these tables know based on the treatment duration in the humans. So what is the corresponding equivalent or corresponding treatment duration in animal study is needed in order to develop either NOAL or MTD that can be used for regulatory filing. So similarly you need to identify if the drug is given in humans for 8 weeks, what is the equivalent animal study that need to be done. 8 weeks means what? 8 is the 56. 56 means from 2 months study. At least 3 months study you should have. Okay. With respect to non-critical route, please go and read that table in detail. So those who are working in drug discovery or IND submission, for them this type of studies is very important to know that in detail. Next is a shift of oxygen curve, that is high temperature, high carbon dioxide and low pH. So one question on oxygen dissociation curve was asked. So please read in detail. Next question was on LD50 and ED50 calculation. Similarly, they might give you a dose response curve and they might ask you to calculate LD50 and ED50 value. Okay. And what is LD50? They will try to confuse you. They will give you LD99 by ED1 or LD1 by ED99, something like that. They will give you four options will be almost nearby, so you will not be able to identify the right option. It will confuse you. Don't get confused. Stick to understand what is LD1 by ED99 or LD50 by ED50. What is margin of exposure? What is margin of safety? What is the formula for these? What is therapeutic index? What is therapeutic exposure? Right. Next, in continuation with this DABT 2022 list of questions based on memory from the previous DABT students, the other topic was in vitro and in vivo genotoxicity study correlation. So one question they will ask you about in vitro in vivo genotoxicity correlation, right? Next is route to administration, biotechnology derived high molecular weight compounds. So this is with respect to ISH S6 guideline. So for S6 biotechnology derived compounds, what are the route of administration in the preclinical you have to do, and based on the molecular weight and size, okay, what type of studies need to be done. So you need to read ISH S6 guideline in detail. The next topic was threshold concept in graph and doses. So in the previous slide, as I discussed, no, dose response curve, threshold concept, threshold dose, type of graph, doses, all these things they will ask. Next topic was ISH Q3R2, impurities and qualification. So one question with respect to impurities you will get. The total ISH Q3, 5 guidelines are there. Q3A, B, C, D and E. E is for extractable and leachables, D is for elemental impurities, C is for residual solvents, and A and B, A is for drug substance, B is for drug product. So ICH Q3 are two. Okay, impurity qualification. So here different types of sources of impurities are also been listed. So this is a favorite topic, one of the and interesting and very much under stringent scrutiny by the regulators. So they will ask you one question with respect to this. Next topic was type 1 and type 2 error. So those who are working in research area, no, uh, for them you will be knowing H naught, H0, that is a null hypothesis, type 1 and type 2 errors. So after application, so um, what are the observations which can be avoided based on the classification of this of this error can be done. Next topic is case control and cohort studies. In continuation with epidemiological studies, they will ask you one question with respect to retrospective case control cohort studies. Okay. Next is another topic was with respect to gray literature data represents. I do not know much about this one. I also got this list from the LinkedIn. So please go and try to understand what is gray literature. Next topic was positivity and negativity data representation. How the positive results as well as negative results need to be represented. Next is antidote for methemoglobin. So methemoglobin we have seen in the previous slide. No, we need to know similarly as antidote for acetaminophen was there. So make a list of the drugs which and their antidotes, commonly asked antidotes. But you will not. This will be a direct question, so you will not lose any mark in this one. Next was question with respect to chloroquine toxicity. Maybe in 2022, hydroxychloroquine or chloroquine were the topic of interest at that time. So probably they might have asked one question with respect to chloroquine toxicity. They might ask you about the exams. They might ask you about the symptoms or the mechanism by which it is causing toxicity. Next topic was about first order kinetics. So they might give you some graphs and they will ask you about first order, second order, third order, second order. You need by based on that graph you need to inference which type of kinetics it is following. Next topic was about two compartmental model, biphasic absorption elimination related. So one question with respect to absorption of a certain chemical they will ask. So in this they had one compartment model is there PK. If you see two compartment model is there, so they have asked with respect to biphasic absorption. Okay, biphasic means probably first it might be slow absorption, then rapid absorption, something like that. So you see, read in detail. Next one is and try to remember the examples also. For first order kinetics, commonly examples of drugs, pharmaceuticals are there. If there are some examples with respect to one compartment model and two compartmental model, please try to remember those example, classic examples. Brain and testis organ weights are not affected with test item toxicity. Probably they might have given you some example and they said that these are the two organs which are not affected. We need to identify that chemical. So these might be some classical this thing based on which they will frame such questions. Next question was about chi-square test and t-test. So statistical one question with respect to either statistics, biostatistics you will definitely get. So they might give you a case study and they will ask you to for what type of statistical test need to be applied. Okay. Next one is aquatic. One question was with respect to aquatic toxicity. Yes, you will get some questions with respect to daphnia or like EC50 in that LC50 ecotoxicity, environmental toxicity you will get. Another question was with respect to buconic acid excreted in low exposure of benzene. Okay, so benzene ka metabolite may be muconic acid. So please go and read it in detail. Benzene, what is the target organs for benzene toxicity? What is the mechanism? What are the toxic metabolites which are formed? Next question was on proximal tubule toxicity in NSAIDs. So NSAIDs, very bigger classes is there, COX-1 and COX-2. So what are the class effect of these NSAIDs? Okay, so whenever they are affecting kidney also, which part of the kidney? Are they affecting nephron? Are they affecting glomerulus? Are they affecting PCT, either distal convoluted tubule or loop of Henle? We need to identify. So in my one of my videos, I have already discussed in class and also they have very clearly given. You have pictures which part of the kidney or nephron is affecting, and they have given you some list of examples also, list of chemicals as well as list of drugs they have given. So try to remember those pictures, those examples. Next is one question on CCl4 and that is carbon tetrachloride and ethanol toxicity. So in continuation with the second chapter, that is principles of toxicology, one question you will definitely get by giving examples about additive effect or synergistic effect or potentiation effect. So they will give you an example of one or two chemicals, two chemicals, and they'll ask you like this: which of the following, or they'll give you one, two, three, four options. It may be synergistic, additive. You need to identify. If you don't know the examples, though you might know the meaning of what is synergism, what is additive, but if you don't know the mechanism of this, how this individually these chemicals are affecting, how they're enhancing or decreasing each other's toxicity, then you will not be able to address that question. So remembering the examples along with these definitions is also very important. That you will definitely get in principles of toxicology chapter is the lesson. And one more thing, this is a classic example. The repetitively they are asking CCl4 ethanol toxicity or benzodiazepines, or some other drugs. And they are rifampicin, rifabutine. So like that, some questions are there which they will rifampicin or CYP enzymes or grape juice with some other drugs. They will definitely ask such questions. Next question was on NIOSH. With respect to NIOSH, in 2024, they have come up with this a new updated list of drugs where there is a reclassification of table 1, table 2, as well as table 3. Some of the drugs have been categorized as teratogenic or carcinogenic. So you have to remember those examples. So please read in detail NICE 2024 list of drugs. So if you see the recent 2025 FDA audits in the pharmaceutical industry in India, whether it was Sun Pharma or I don't know mention pharma in India, I'm saying. So one of the observations what FDA have seen that they were checking for the NICE listed drugs. How the pharmaceutical industry or scientists are handling this NIOSH listed drugs they were looking. So try to remember. If there are some important classic examples, read those things. Okay, try to remember those table 1 to 3 examples listed under NIOSH 2024. Next question was on FDA, CER, CLA, CERCLA. So whether it is CERCLA or Superfund, FDA has one program. Okay, where that program is meant for, that organization is meant for how to handle waste, any chemical spillage is there, or disposal of the unwanted chemicals are there, whether it has to be dumped in the soil or it has to be discarded in the drums or in the water bodies, that is oceans. So CERCLA is a body which takes care of the this thing. Particularly I will say the product stewardship. If a chemical is produced by certain industry, how they are disposing that chemical, how are they handling that chemical, whether that chemical has been loosely left into the environment. So all those things, CERCLA or Superfund is the organization which will be taking care. So there were historically lot of incidents are there because of which that made the FDA to bring into picture of this organization, CERCLA or Superfund. Since the exam is conducted by the FDA people, that is in the US, that is DABT is from America, so they will always try to bring these type of questions also in their exam. So try to read it first, understand what is CERCLA, what is Superfund. This CERCLA or Superfund are both same. So for waste disposal, this thing is there, no? What are the organizations? So which type of organization is handling which type of this thing, you should know. Pesticides ke liye what is there? FIFRA is there. The Toxic Substances Control Act is there. What is there? Then we have this one, circular superfund is there. You have to remember US EPA is taking care of what. Okay, PPRTV is there who's taking care of what. These are the organizations from the FDA which have various applications for various types of product. EFSA is there. Now, EFSA is European. I mean, the food products के लिए food organization FAO, WHO, FAO is there. So like that you need to know. Next topic was on the carcinogenicity category based on M7 guideline. So coming to ICH M7 guideline, no, please read in detail. In the M7 guideline, you have 5 classes of drugs. Are class 1, 2, 3, 4, 5 are there. So based on the utilization, application of this software from the DEREC or LASA, or based on the availability of the carcinogenicity studies of those chemicals, with if they are, and if they are listed under the CPDB, that is carcinogenicity potency database, you will have a TD50 value from which you can calculate an acceptable intake. So like this one question you will get M7 ka. So they might give you some examples. They'll ask you what category it belongs, what class it belongs, whether 1, 2, 3, 4, 5. That you should be knowing. So these are the class 1, 2, 3, 4, 5. Read M7 guideline in detail. Next topic was on toxicology report, study report, findings and adaptive changes. They might give you a question. They might give you a study in that a couple of sentences will be there about the study results from a study from a preclinical study like 28-day study or 90-day study. They will give you some doses and they will give you some observations. Then you have to identify those observations. Which of them is adaptive change, which of them is adverse change, which of them is treatment related, based on which they will ask you to identify the derive the noel also. So options will be like that. So those who are study personnel or study director, they will know in detail. Of course, those are in the risk assessment business also. They will be also knowing this type of topic to address. Next one is question was on sister chromatid assay, that is micronucleus test, comet assay, and UDS assay. As we have seen in one of our previous slides, sir, one question on genotoxicity battery of test. So there will out of we have some 40 plus or 50 plus gene tox assays are there, mutagenicity, clastogenicity assays are there. In that also important questions, no micronucleus test, AIMS assay, chromosome aberration assay. These are important. In addition to that, in the options you might also come across sister chromatid assay, comet assay, unscheduled DNA synthesis assay. So you need to know what type the DNA damage assay and what does it influence, what type of damage, whether you can know anagenicity, whether you will come to know about clastogenicity, whether you will come to know about the frame shift mutations, like that. Which essay is teaching us what about that chemical type of damage that you need to remember. Next one topic was on reproductive toxicity, that is pre-implantation loss and post-implantation loss. Okay, so there are few formulas when you look into ISHS5 guideline. No, there will be how to calculate number of liter size or a number of pregnancy. There are some formulas are there. Post-implantation loss, pre-implantation losses. So remember those. One of the formulas they will try to confuse you in the in the question. All the formulas, no, they will be nearby. You will not be able to identify. So read in detail ISH S5 guideline in detail. Next question was on duration of dosing and days of drug administration to mother in pre and post in natal developmental toxicity. In continuation is ISH S5. They might ask you about pre and postnatal developmental toxicity also. Next question is statistical significance related to reproductive toxicity based on both pregnant mother and litter. So you need to understand what is the difference between biological significance and statistical significance. Okay, so there is a difference out of which you will be considering in your final conclusion of your report. Next is why biological data is more important than statistical significance, similar to the previous the one. Then one question was on sulfur dioxide, London smog, photochemical smog, and London smog. Again, these two questions also I have already covered in one of my videos. So they might give you some which of the component is not a or for photochemical. They will ask you the formula for photochemical smog or London smog, ozone, sulfur dioxide, carbon monoxide, nitric oxide, peroxynitride, like that. And one question was on tidal volume, functional capacity, and vital capacity. With respect to respiratory toxicology, so they will ask you about these parameters. So whenever you are doing a inhalation toxicology study, you need to know about tidal volume, functional capacity, and vital capacity. Okay, there are certain formulas are there. So please read OECD guidelines with respect to inhalation toxicology. At least one study, one guideline, one reading you give, you will come to know about the formulas. It is very difficult for the people who are not already working on inhalation toxicology, but try to know at least give one or two readings about this particular guideline, because of which by seeing the options at least you will be able to identify the right answer. Okay, next topic was the nanoparticle and absorption. So key important thing that you have to remember, as the surface area decreases, as the particle size decreases, surface area increases, so absorption increases. So this is the blind rule you need to remember. So in case of nanoparticles also, the reason why people have gone for the particle size reduction is to increase the surface area, to increase the absorption, so more bioavailability will be there. That was the but because of the extremely small size of these nanoparticles, the nanoparticles by itself they are causing toxicity. So that you need to read. So we have a separate chapter in the lesson with respect to nanoparticle toxicology. So try to read what is the size of the nanoparticle and which part of the region in human body or which part of cells are getting damaged by the reduction in the size of the particles that you need to, because these particles, even though they are nanoparticles, these particles may aggregate and form a bigger structure. They might become microparticle, I guess. So you need to read it in detail. Okay. Then next, we had topics with respect to impaction of dust load in lung. As I told you, lung particle size, which area it is going and deposit, that you have to know. Next is 15 microparticle deposit in pharynx. So you have to remember the particle size, part of deposition. Next topic was oil soluble at 5 mg per liter and forms layer over water surface in fish toxicity study. LC50 at higher concentrations can be determined or nor up to 100 mg per liter with all mortality. See, what is important here is that they will ask you with respect to hydrophilicity, hydrophobicity. If you see lake or pond or any water bodies, based on this, whether density, whether this particular oil will retain on the surface, or with how it is impacting the ecotoxicology, the ecosystem, means fish will die or like that, some questions you will be getting. Okay. So LC50, EC50 with respect to daphnia or fish, they will ask you one question. Next topic was glyphosate poisoning related, that is pesticides. This again related to herbicides or pesticides. Remember what are the important herbicides, important pesticides, their mechanism of action, right? And if they have any classic symptoms or adverse effects by which you will be able to identify, yes, this is the herbicide or this is the pesticide which is causing this one. So in that options you will come to know classification of the pesticides and herbicides, etc., based on, right. Next question was on carbon disulfide, so CS2. So one question on CS2, how the cascade, the mechanism you need to read. Drosophila and fish toxicity, which is similar to this one. Then brevitoxin, chrombytoxin, puffer poisoning, as I said, no. One question with respect to uh, coming to animal toxins, they might ask you about the fish and the type of poisoning. They might ask you about regarding snake or poisonings also. There are some list of commonly animals are there and their toxins which are produced by these animals. So one question you will get on that one. Then next is about microcystin toxicology mechanism. Please read in detail the particle size, how they are forming, how they are disrupting the membrane, right? Then ARH receptor binding and toxicity. ARH, aryl hydrogen receptor, because of highly reactive, they will react with the your cell membrane. Some reactive oxygen radicals might form. So please, how this AR their mechanism you read in detail. Next about cadmium poisoning. So there is a separate guideline ICH-Q3D with respect to elemental impurities. So one question you will definitely get about metals. So you need to know about what type of metal, what is the symptoms, what is the toxicity which is causing, right? Remember in the periodic table you have 100 plus elements are there. Some of these elements are already available in our body, some of the elements are abundantly available in the earth crust, okay, in the atmosphere. Some of the elements we are on a day-to-day basis we are getting exposed. Some of the elements might also have some pharmaceutical applications. So some of the these element metals or elements might come in our water bodies also. Okay, so we need to, but we need to limit those. For example, lead levels are there. So lead is there, arsenic is there, by for which by default they have established certain safety limits. That is there in the case of ICH-Q3C guideline. In the ICH-Q3C guideline, they have already given PDE levels for, they have given class 1, class 2A, 2B is there, class 3 are there, and metals to be avoided something are there. Please read in detail. So one question with respect to metal toxicity. Metal toxicity in the Clayson, again there is a separate chapter is there. Okay. Then next we have another topic, CYP2E1 related metabolism. So one question with respect to CYP450 or CYP2E1, they will ask you. Okay. So what are the drugs? If you have, there are some examples are there. Please remember, try to remember this CYP2E1 or CYP450 also. Okay. Then next we have in continuation we have glycolic acid conversion to glyoxylic acid and the rate limiting step. If there are some of the important with respect to biochemistry, I'm saying, if there are some of the important chemical reactions are there, then for that chemical reaction we need to remember rate limiting steps. So one of the options they might give in the case of glycolic acid conversion to glyoxylic acid, they're asking for rate limited step, right? Then there is about another question is with respect to glycol ether, methoxyacetic acid. So in the case of glycol ether, diethylene glycol ether, diethyl. So one of the again glycol toxicity, either DGME or EGME, favorite topic for the examiner. So you need to remember the toxic metabolite which is formed and which is responsible for causing the toxicity, whether it is causing teratogenicity or whether it is causing kidney toxicity, that you have to remember. And there are recent examples are there because of which these solvents, peg solvents, there are withdrawals from the market, certain syrups, cough syrups. So you need to remember those cases. Okay. Then we have another question with respect to toluene toxicity and effects on brain. Okay, how the solvents, how they are affecting your CNS. Then cocaine suppresses means recreational drugs, how they are affecting your serotonin reuptake mechanism. Okay. Then we have another topic on chronic kidney injury related. So you need to, when it comes to kidney toxicity, you need to understand what is acute kidney injury and chronic kidney injury. And you have to also remember list of examples also. Classic examples are there with respect to AKI as well as CKI. Try to remember those examples. Next one is dietary toxicity depends on feed intake and actual drug dose in feed. Okay, please read EFSA also with respect to this one. Then you will understand this feed intake, the dietary conversions, right? Next one is renal papillary toxicity. So coming to kidney, as I said, no, which part of the kidney is responsible for what type of toxicity? You have to remember papillary, calyx, nephron, glomerulus, all those things. Next one is immune mediated stress induced immune mechanism. So one topic is in our syllabus is about immunotoxicity. So one question where they have asked with respect to immune mediated stress induced immune mechanism. So mechanisms you have to remember. Next here here it is not chemical agent, it is stress. Okay, then we have PTH, parathyroid hormone released calcitonin related one question they have asked. So we have to remember this pituitary parathyroid hormone. Okay, endogenous hormones are there, no? Their mechanism, how they are causing, um, how this is released, that you have to remember. Next question was with respect to micro sampling, avoid animal stress. So how by using, here you need to remember about 3R principles. So we have reduce, refine, recycle. No, so in the case of refinement, there should be refinement is nothing but there should be changes in the process, in the way we are carrying out a certain experiment. So in that micro sampling, if by using less invasive techniques and by using this micro sampling technique, if we are able to get the same results, then what we were you doing macro sampling, then that will come under the refinement type of principle, 3R principle. So that will cause a less stress to the animal. So for that reason, you have to one of the question might they have asked on this. The next about clinical trial guidance. So please read E6, ICH E6 guideline in detail. Okay, those who are working in clinical trial and clinical toxicity, they will be knowing. The next another question was on specificity, prevalence, non-negative, false positive. So you will get some kinds of case studies. They will ask you about positive, false positive results, false negative results. One question. So please read these terminologies in detail. Next question was on ANOVA. How like three or more groups to consider. So type of test. In the previous slide, we have seen a T test was there, chi-square test is there, ANOVA test. They might give a case study and they'll ask you what type of statistical test need to be inferred in the case of, in this case. Okay. So next question was on dominant lethal assay. Okay, that again dominant lethal assay maybe with respect to ISHSP or genotoxicity, and repeat dose 90-day requires as support for phase 3 clinical trials. So again this is with respect to ISHM3 guideline. Blue light in infants helps in bilirubin synthesis. Please read this one. Okay, then plasma protein binding and volume of distribution VD with respect to kinetics. Then warfarin protein binding and drug distribution. So anticoagulant drug, bone marrow toxicity. Then cell mechanism of death, that is necropsis, apoptosis, necroptosis, ferroptosis. Please read these terminologies, don't get confused. Then one question in continuation with risk assessment, they will ask you about hazard index and hazard coefficient. They will ask you to calculate also, they will give you certain numbers. certain case study, and they'll ask you to calculate HQ ratio. They'll ask you to calculate risk characterization, risk assessment, dose response, risk assessment, exposure assessment, and risk characterization. In continuation, this you also read about risk mitigation, risk communication, right? Then next topic was on occupational exposure. In this case, they will ask about engineering, employee measure, others are not in place in the main. So, what are the alternatives? PPEs can be used. So, one of the trending topic with respect to these FDA inspectors is about occupational toxicity. So, they are keen regarding the worker safety and the scientists who are working with these chemicals. So, what are the PPEs that need to be applied, and what is this occupational exposure? Okay, what are the rules of exposure? And what are type of safety limits that can be, like occupational exposure limit? So those also you read in detail. Next topic and question was on in vitro test, not developed for corrosive substances. So we have two types of things: skin irritation is there, skin corrosion is there. So try to read the difference between. One is skin irritation is reversible, whereas skin corrosion is irreversible. Once the tissue has been subjected to corrosion, that is a irreversible damage. Okay, so for them there is no need of doing any uh, or that is eye toxicity, and you will not be doing any sensitization also in that case. So read these terminology like a skin irritation and skin sensitization, right? And corrosion substances also. Next one is historical control data is useful for verifying the control data and test items to support and any ambiguity. Very important. In the GLP system or in the GCP, that is clinical trials, historical control data plays an important role. There are certain observations. If they are in line with historical control data, those observations can be cannot be considered as adverse effect. Okay, that will help you in deriving the NOAL or MTD. Okay, so these are the questions which I came across. This list of questions I got it in the LinkedIn, so I have just listed out so that you'll remember. These questions were based on the memory of the DABT students who appeared in the 2022. Okay, thank you so much. Be curious. For any question, please comment. Okay, like, subscribe, and follow my YouTube channel, Toxicology on Board. Thank you so much, and all the best for your exam.